It's kind of wild to think that the same basic science that helps us bake a loaf of bread is now on the verge of curing genetic diseases. I mean, that's biotechnology for you, an ancient practice that has absolutely exploded into a modern powerhouse. So how on earth did we get from a simple bakery to a cutting-edge biology lab? Well, let's dive in and break it down. So here's our roadmap. We're going to start this journey way back in ancient history. Then we'll zoom right into the supercharged crops on our plates and the revolutionary medicines in our hospitals. After that, we're going to get to the very code of life itself. And we're going to wrap it all up by asking the biggest question of all. Just because we can do all this, does it mean we should?
Okay, let's start at the very beginning. The word biotechnology might sound super modern, but its roots, they go way, way back. And to really get a handle on its power today, you've got to see the massive leap it's taken from just, simple fermentation to incredibly precise genetic engineering. Now, what's so fascinating here is the fundamental shift it shows. You see, for thousands of years, old biotechnology was all about using natural processes. We didn't really understand, how does yeast make bread rise? We just knew it worked. But modern biotechnology, oh, that is a whole different ballgame. It is all about precision. We've gone from just working with organisms to literally becoming the editors of their genetic code. Okay, so where's the first place we see this new power at work? Well, look no further than your dinner plate. The first, and maybe the most widespread application, is right there in our food supply. Biotechnology is completely changing the game when it comes to how we grow crops, making them stronger and way more productive. And the star of this agricultural show is the genetically modified, or GM, crop. The whole idea is actually pretty simple, but wow, is it powerful. Scientists can find a useful gene in one living thing, let's say a bacterium, and then insert it into a plant to give it a very specific, desirable trait. Seriously, you can think of it as giving plants literal superpowers. And here are two perfect examples of these superpowers in action. Take bee cotton. Scientists found a gene in a soil bacterium that makes a protein that's toxic to certain insects. So they put this gene into cotton, and voila, the plant itself becomes poisonous to pests like bollworms. This drastically cuts down on the need for chemical pesticides. And it's a similar story with making crops resistant to herbicides. It just lets farmers control weeds way more efficiently. But these superpowers aren't just about making farming easier. No, we're talking about tackling massive global challenges here. Biofortification, like adding vitamin A to rice, can literally help fight malnutrition across the globe. And in a world dealing with climate change, creating crops that can survive a drought or grow in salty soil, well, that's not just a nice to have, it's absolutely critical for our future food security. All right, let's switch gears. We're moving from our fields to our hospitals, into what is honestly maybe the most life-saving part of biotechnology, medicine. The same principles of genetic engineering are creating revolutionary new treatments and ways to diagnose diseases.
A perfect example of this? Insulin. Before biotech, insulin for people with diabetes had to be taken from pigs and cows. It worked, sure, but it wasn't a perfect match, and it could cause allergic reactions. But today, scientists have put the human insulin gene into bacteria. These bacteria then become these tiny biological factories, just churning out a limitless supply of pure human insulin. It's safer, it's more effective, it was a total game changer. And in the fight against cancer, biotech gives us this incredible three-pronged attack. First up, early diagnosis. Genetic biomarkers, like the famous BRCA1 gene, can help us spot the risk of cancer years before it ever develops. Then, you've got targeted therapies. These are a world away from old-school chemo. These drugs are designed to attack proteins found only on cancer cells, leaving healthy cells alone. And the final piece of the puzzle? Immunotherapy, where doctors can literally engineer a patient's own immune cells into super-powered cancer killers. So, all of this, it brings us to the absolute cutting edge, the most powerful, most revolutionary tool in the entire biotech arsenal. We're talking about the ability to not just add genes, but to edit the existing code of life with pinpoint accuracy. Welcome to the world of CRISPR. So here's the absolute best way to think about it. If you imagine the billions of letters in an organism's DNA as a giant book, CRISPR is like the find and replace function in your word processor. It lets scientists find one specific genetic sequence, a typo if you will, and just fix it. Okay, so how does it actually work? Well, the most popular version is called CRISPR-Cas9, and it has two key parts. First, there's a guide RNA, which acts like a biological GPS, homing in on the exact spot on the DNA we want to change. Once it locks on, the second part, an enzyme called Cas9, acts like a pair of molecular scissors and just snip. It cuts the DNA. From there, scientists can either delete the faulty gene or slip in a corrected version for the cell to use. I mean, the potential here is just breathtaking. We're not just talking about treating diseases like sickle cell anemia or cystic fibrosis. We are talking about potentially fixing the broken gene that causes them forever. It could be used to edit our immune cells so viruses like HIV just can't get in. And back in our culture, you could create strawberries that stay fresh for weeks or tomatoes that taste incredible, all without adding genes from another species. But with great power comes great responsibility. You know how it goes. And with the ability to rewrite the very code of life, we have to hit pause and really think about the profound ethical questions this raises. We've gone from simply observing life to harnessing it, and now to actually rewriting it. And that path, it forces us to confront this one ultimate question. Where do we draw the line? Okay, so the concerns really boil down to three big areas. First, there's the safety issue. I mean, do we really know the long-term consequences of making permanent changes to a genome? Second is equality. If these therapies are incredibly expensive, are we at risk of creating a genetic divide between the rich and the poor, a world of so-called designer babies? And finally, you have this massive philosophical and ethical concern about humanity's role, this fear of playing God and maybe causing unimaginable consequences we can never take back. So as we've seen, this powerful science is already all around us, whether it's cutting down on pesticide use in our fields with things like Bt cotton, or creating safer mass-produced medicines like insulin, or even just offering the hope of one day curing genetic disorders with CRISPR. Its impact is absolutely undeniable. So in the end, biotechnology has handed us the pen. We can now write the next chapter of our own evolution. The science is moving at lightning speed. It's giving us the how, but it's up to us, all of us, to have the conversation about the why and the if. It's a tool of immense promise and, yeah, potential peril. And the story we choose to write with it, that's going to define our future.